for functional alleles. So the second assertion then was that wild species hold promise for chickpea improvement. But there was a caveat, and that was that, at least according to our assertion, and I'll show you some data to support this, that the existing germplasm collections were inadequate to fully harness the natural adaptations. That's not to say that the existing collections aren't useful, but they're probably not useful for a systematic deconstruction of function from natural populations. To give you a sense of that, so what are the factors that have limited the use of wild material in the case of chickpea? The first is that we lack a systematic understanding of natural variation. We could invest as much as we want in understanding the current world collection, but it's not been sampled in a way that allows us to really systematically understand natural variation, to be sure that we've captured it properly. So that's the first problem. There's no systematic approach to having collected that natural variation. A second important issue that has limited the use of wild material is there are large phenological differences, particularly response to the environment that affects flowering and rates of development between cultivated and wild. And this photograph is taken here at ICRISAT in a project that where we collaborated with Rajiv Varshney and Porn Gar. And what you can see on the left-hand side here are a series of different genotypes of cultivated Cicer eritinum or chickpea, interspersed with a series of different genotypes of wild material, particularly Cicer reticulatum. And you can see that the wild material is growing prostrate on the ground, and it's not flowering efficiently, at least in this particular experiment. So there are very large developmental differences. And you couldn't reliably compare phenotypes in this field experiment between these eritinum genotypes and those reticulatum genotypes because their phenology is too far different. Now the interesting thing is if you take genotypes of Cicer reticulatum, this material here, and you grow it under different environmental conditions, now it grows much more like cultivated material. So it's very, very sensitive to environmental conditions, and this is one of the things that we're going to try to address. But it complicates phenotyping within the wild material. A third thing that has limited the use of wild material is that it's a lot of effort to make crosses and do the phenotyping. And so there have been no comprehensive attempts to comprehensively integrate wild material into populations in which the genetic potential of that wild material could be studied. And the reason is that it's difficult and time-consuming to conduct these things. So the project aims to address each one of these issues, to rationally characterize wild material, to construct populations that remove extreme phenological differences from the equation, and to conduct massively parallel population development so we can begin to survey the function of natural alleles. So this is sort of the project in a nutshell then. We're working with two wild species, Cicer reticulatum, which is the immediate progenitor of Cicer eritinum, and a sister species, Cicer acinosperma. These two species occur in southeastern Turkey in particular, in Mesopotamia. And the goal is to identify populations, characterize those populations, and transfer the wild alleles that are present in these populations into a cultivated context so they can be assessed functionally. And as I mentioned a minute ago, the project combines ecology, genomics, germplasm development, pre-breeding, modeling, and finally, either in this project or after the project is finished, breeding. So we're moving from the wild environment into the agricultural environment with wild alleles. Now the project is funded primarily by USAID. I've already introduced that. There's a corresponding project funded in Australia by the Grains Research and Development Corporation, a project of equal size as the USAID project, and they're going to focus on phenotyping the material that's been identified and for which populations have been developed or are being developed. And there's a third project of smaller size but still large in scale, funded by the Canadian government at the University of Saskatchewan. So there are three separate projects, the main USAID project and then two allied projects funded in Australia and Canada. In addition to those projects that focus on identifying, understanding, and then trying to utilize wild alleles, there's a fourth project that's funded by the National Science Foundation, and this project has a more basic focus. The focus of the National Science Foundation project is to look at nitrogen fixation, and particularly the impacts of domestication on nitrogen fixation. And so the NSF project is looking at this organ, which is the nodule, of course, on a legume root. It's the site of nitrogen fixation in which this important biochemistry occurs and atmospheric Dinitrogen is reduced to ammonia and then eventually to organic forms. An obvious competitive advantage of legumes in natural systems, an important component of them in agricultural systems. So like the USAID project, where we're looking at climate resilience, the NSF project is also interested in this gradient, but asking what happened to nitrogen fixation and nitrogen-related processes during domestication. And I'm not going to talk about this in any detail except to say that there are large phenotypic differences between wild cicer species, particularly reticulatum, and its derived cultivated species, Cicer eritinum. There are differences in the way the species respond to soil nitrogen. There are differences in the specificity of the species for symbiotic rhizobia within complex soil environments. There are changes in the way they allocate carbon based on nitrogen availability. And there are shifts in the number of nodule organs that are formed on the plants. And we hypothesize, but have not yet shown, that there is reduced efficiency for nitrogen fixation in cultivated species. So in essence, we have the USAID project focusing on climate resilience for improvement, and the NSF project focused on the discovery of traits related to nitrogen fixation in wild species that also could potentially be migrated into cultivated material. And that project also is relatively new, although we have more data there. So both the projects anticipate using wild material, and so there's a fundamental foundation that's important to both projects, and that is collecting and understanding the functional diversity of wild species. So the first speaker in the conference talked about genomics in a large context, and he made a point that I thought was interesting. He basically described a situation where, let's say you're looking at gene expression or maybe methylation. We look at it in a static way, usually one or a few time points. We take a picture of the organism. But he made the point that what you'd really like to understand is the dynamics of those processes over time, not in discrete time points, but in a continuous series. And to me at least, it described how far we need to go in order to be able to understand system function in a complex way. But he was talking about single cells or maybe individual genotypes of a species. In our project, we turn the equation upside down and ask, what about the functional consequences across landscape scale distribution? So ask yourself, how would you sample in a rational way and then dissect the genetic potential of a species that spans 60,000 square kilometers, which is the region that's encompassed by Cicer reticulatum and its sister species, Cicer iconospermum, a relatively small geographical region. Those two species actually have a very narrow distribution relative to many wild species, but it's still a big area. If you start driving around in the car trying to find populations, it's not a simple thing. And so the project focuses then on this southeastern region of Turkey. The majority of Turkey in this map would be up and to the left, so northeast. But the distribution of the wild species is between primarily between the Euphrates and Tigris River in a region called the Haran Plain. It's bounded by, it's a relatively high plain, about 2,000 feet or about 700 meters, and it's surrounded by mountains of increasing elevation. And this is where the species occur. Immediately south of this is Syria, where the species doesn't exist, but you couldn't sample right now, even if it did, and Iraq and Iran, that for our purposes were off limits in terms of sampling. But most of the species, in any case, occurs within Turkey. And so we wanted to sample there, and again, we're hunting two types of species. The species that precede the formation of Cicer eritinum, the cultivated species, Cicer reticulatum, the immediate progenitor, and a close sister species, Cicer iconospermum. It turns out, and I won't come back to this later, so I'll describe it now, that after we did the sampling, we sort of developed a search image for where the species would be distributed, that iconospermum exists primarily in the plains, and reticulatum, the species we're most interested in, exists in the foothills and mountainous regions. So our approach to finding the functional diversity of those species is to take an environmental-based approach. Basically, the idea is we assert that if you want to understand the functional attributes of the species in the natural environment, you have to sample the environmental extremes across which that species exists. And so before we went in and started collecting the species, we modeled the environments using GIS data and global data about climates to come up with ideas about where the species might be distributed. And then we hopped in cars and spent 2 months surveying those areas and expanding those. The goal in surveying this diverse set of environments is first of all to collect as comprehensively as possible, but to make sure we span the environmental extremes. Because the traits that we want to have phenotypes for are particularly tolerance to climate. As you'll see that we sampled altitudinal gradients, which is the main way in which we sampled environmental gradients. We sampled extremes of rainfall. We sampled differences in the seasonality of rainfall, differences in soil types, and so on. And in doing so, we expect to find populations that are adapted to those differences. So we're interested in climatic adaptation, and in addition, climatic adaptation for nitrogen fixation, because abiotic stress is a significant limiting factor for nitrogen fixation. Within these populations, we also expect to find genes for disease resistance, and a number of our partners have interest in particular pathogens of chickpea that also occur in wild natural systems. We're interested in soil type adaptation, nutrient density, a range of different traits will be phenotyped in the material. And I already mentioned that we combine a diverse set of disciplines to try to approach this somewhat complicated problem. Let's see, there we go. Okay, so I mentioned, so we've got a search image. We have a general idea of where the species is distributed. We spent 2 months in southeastern Turkey, and we did this basically in 2 trips in several different trips, but each trip we had 2 teams, somewhere between 10 and 15 different individuals. And we just drove around and started identifying sites where the species was located. We spent 2 initial trips in May identifying the sites where the species was before they set seed. And we marked and tagged all the plants that we could find. And then we went back on 3 additional trips over the remaining about a month and collected the seed as it matured. And what we ended up with was a large collection of material across the known geographical distribution of both Cicer reticulatum and Cicer echinaspermum. We sampled 23 different sites. Now, I'm not referring to these as populations. These are simply geographical locations. They may or may not be some number of distinct populations, which we can only determine by looking at the characteristics of allele frequencies within those sites. So 23 different sites. 3 of those sites ended up being a species called Cicer bitigum that we're not interested in, so we have 20 sites that represent primarily Cicer reticulatum and a few of them that represent Cicer echinaspermum. Each time we went to a site, and these are 2 different sites, a reticulatum site on the right and an echinaspermum site on the left, as it turns out. Each time we would go to a site, and this was in May, the goal was to find every plant at that site, not just some plants at the site. And every time we found a plant, we put a flag. It doesn't matter whether they're 6 inches apart or 200 meters apart. Everything is tagged. Every plant is given a GPS coordinate, and for as many plants as possible, we collect soil underneath that plant. So now you end up with a description of the spatial scale distribution of those individuals across those 23 different sites. Each one of those individuals will ultimately be sequenced, and I'll show you a little bit of data about that. So what you understand at that point is the distribution of genetic diversity across sites. Are individuals that are close more similar to one another than individuals that are far apart, for example. We also went back and embedded microsensors in the soil that would give us hourly readouts of humidity and temperature. It's a seed's eye view of the world. So we understand now not just regional variation that we can get out of the databases, but now we understand microsite variation to which the plants are evolving, because the site is heterogeneous in terms of the factors that influence that exert selective pressures on the populations that exist there. So we tagged everything, took GPS coordinates, collected soil. We're collecting rhizobia in parallel from all of these sites. And then we went back later and collected seed. And we brought that material back to the laboratory. So what did we end up with? About 840 plants as DNA, about 400 plants as seed, about 150 soil samples, and so on. So a range of different attributes. Now I should say, also related to the first speaker of the session, where he talked about the function of the microbiome and soils being relatively inaccessible environments. We're going back in a few weeks and we're going to sample the microbiome of the species in situ, again using this spatial scale analysis, looking both at endophytes and epiphytes, particularly bacteria. So each of these sites will become laboratories for trying to understand evolution, but we expect them to be also the location of genes that are involved in adaptation to these different environmental gradients. Now, so what have we sampled in terms of environmental differences? Well, this just shows that we've sampled altitudinal gradients spanning about a 2.5-fold difference. And the highest elevation is in the far eastern region in a town, near a town called Cernak, for example. So we have altitudinal gradients, and we know that altitudinal gradients are going to give us environmental gradients because we don't have a large distance that we're talking about, and most of the environmental differences will be driven by altitudinal differences. So we can also begin in a very preliminary way to model the environments at environmental levels, and so we can collect information from global repositories. We know that, for example, we have sampled rainfall gradients. We know that with increasing latitude comes decreased precipitation. So those populations are evolving to different levels of rainfall. We know that across the east to west gradient, in fact, there's also a rainfall difference. In this case, it has to do with whether those populations experience rainfall primarily during their vegetative phase or primarily during their reproductive phase. Now remember that each population is adapted to its particular site, so we should expect to see that they have adaptations to these different patterns and availability of moisture, for example. But again, encompassed in these populations will be many, many different differences at the environmental level, not just rainfall, also temperature and a range of other things. So we started to characterize the sites. This is a picture of a typical environment. It's not the only kind of environment where Cicerocynaspermum in particular is found. They often occur in oak pistachio rocklands. There are a wide range of different cultivated species that are native to this part of the world, and pistachio being one of them. And this is where we spent 2 months driving around. Now, part of the story that I'm going to tell you, in fact, sort of has an Indiana Jones character to it. This is southeastern Turkey, and this is a part of Turkey that's been basically in civil war for the last 30 years. And that's where we needed to go to sample the species, because that's where the species is distributed. So we hopped in a car and drove on this dirt road for many days, in this particular case headed north. And we just entered the part of Turkey that was controlled by the PKK forces. And about 3 hours before we entered, they agreed to lay down their arms. So as we were sampling, we met people with machine guns and all kinds of other things. We saw cars blown up by landmines. We spent one morning sampling at the base of the mountain where the Quran says Noah's Ark is located. We had all kinds of great experiences. One group got arrested by the Turkish special forces. But nobody was killed and nobody was hurt. And we ended up with a lot of material. So what are we going to do with that material? I've told you generally, but let me tell you a little bit about the initial data. Remember, it's a young project. So we've got about 1,300 accessions, of which about 150 are a species we're not interested in. So about 1,200 genomes. We want to sequence all of those because we want to use population genetics to infer, based on allele frequencies, for example, the kinds of genes that might be involved in local adaptation. I say might because you just get presumptions from that initial data. So we have to sequence everything relatively deeply in order to get allele frequencies. But we can't afford to sequence right now 1,200 genomes at high depth. So the first thing that we've done is to take those genomes and sequence them at high depth using GBS. We sample about 250,000 genome locations, about 100 base pairs each, using RAD resequencing. And that data is generated, and I'll tell you about it. That data is going to allow us to select from the larger set of material a smaller set of genomes that will sequence at high depth. So we understand the alleles present in the population. We'll sequence 200 to 300 accessions at relatively high depth, and that'll set a minor allele frequency threshold at which we understand the genetic diversity present in that material. And the rest of the genotypes will be sequenced at relatively low coverage using imputation to deduce, based on that minor allele frequency cutoff, the structure of their genomes. And we'll have a description of the genomes and the genotypes that are distributed across those landscapes in the ways that I described to you and that are characteristic of our collection. Then we'll use population genomics and population genetics, for example, to correlate allele frequencies with environmental differences, estimate the nature of selection, look, for example, for evidence of selective sweeps, a wide range of different characters and attributes to be determined from those populations, but none of those are going to be formal proofs of function. There'll be candidates that might be involved in adaptation. Then we'll build populations that will start in the next few weeks. We'll build a nested association mapping panel. It'll have about 5,000 individuals in it. And we'll build advanced back-cross integration lines, where we'll take segments from the wild genomes and integress them into cultivated material. They'll be almost breeding-ready material, carrying small pieces of the wild genomes. And that will be our main laboratory to begin to understand function. Okay, so where are we at? Well, I should say that one of the important sort of enabling features of the project is a project that we had participated in early on. It was led by ICRISAT, and that was the sequencing of one of the chickpea genotypes, a Kabuli genotype by the name of CDC Frontier. And this is a picture taken at ICRISAT. Rajiv was the lead on that project, but that draft of that genome is an important foundation for the analysis that we're doing. So that material came back to Davis late last year. We've germinated the seeds. We have them growing in the greenhouse. We extracted DNA both from the leaf material that we collected and from the plants that we germinated, and we've sequenced it using rad genotyping. As I said, about 250,000 loci in each case. There was some discussion earlier about the cost of genotyping. It costs us about $15 to sequence a genotype. We think when we get into the phase of this, when we're making populations that have high LD, the cost will drop substantially, close to about $1 a genotype. Now, you can take that data, and there are numerous metrics. The first thing you're going to know is how much diversity is there and how many populations are present and how different are the populations from one another, and how does that compare to the existing collections of wild material. And we're beginning to be able to ask those questions. I say beginning because the data is quite recent, but for example, this is just taking the Cicer reticulatum material and a little bit of cultivated here. And you can see, running principal coordinate analysis, that you can divide different groups of Cicer reticulatum. They're not necessarily populations because principal coordinate analysis doesn't tell you anything meaningful about populations, but it tells you something about diversity and differentiation. But we can also run more formal metrics of populations, and we've, in the initial phase, run a program called Structure that basically uses allele frequencies, because at each site we've sampled up to 100 different individuals. So from those individuals, we can come up with good estimates of allele frequencies, and we can compare those within and among sites and ask how many populations have we captured in those 23 sites that we sampled. And the answer is that somewhere between 4 and 10 different populations. And I'm not hand-waving, but in fact, you'll see that probably 4 genetically defined populations, but clearly up to 10 distinct genetic units in the collection. So this is the structure analysis. The first thing that divides out is that Cicer Echinaspermum separates from Cicer reticulatum. Cicer eritinum also separates. The important thing for us is this collection of Cicer reticulatum. There are at least, depending upon the K value that you use, either 4 or 7 different populations of Cicer reticulatum. And that's important. As I'll show you in a minute, that existing germplasm collections sample primarily one of those populations. So we know immediately that we've expanded the genetic potential substantially in the wild material. Most of the current collections of Cicer reticulatum sample within a relatively narrow geographical region. Not all material, but the vast majority of it comes from a relatively narrow region of Turkey around a town called Savur. And we've sampled much more extensively. These are the blue genotypes. A number of sites around here have that population. But there are a range of other populations out west, on the north, and on the east side, particularly as we begin to move into the higher elevations of the mountains. And so one of the things that we'll do this year, we did sampling last year based on using environmental differences to drive our sampling criteria. Now we know something about genetic diversity, and we can say, let's go in and sample those regions and push further where we found novel sources of diversity. So in the next 2 years, first beginning in about 2 months from now, and then a year from that, we'll be back doing the same thing, pushing eastward and northward, sampling more here, moving north and moving west to expand the total amount of diversity and hopefully new populations that are present in our collections. Now, we haven't done any systematic phenotyping, but you might ask, is there any diversity phenotypically? And the answer is there's lots of diversity. If you walk in the greenhouse where the plants are grown in a common garden, you can immediately identify, oh, that group is from Adiman, that group is from Ale, that group is from Cernak. Because there are population-specific differences in growth habits, leaf size, trichomes, pod size, a range of different characteristics. And this just shows one of those characteristics, and that's seed coat color, which may be involved in a process that ecologists refer to as crypsis, but not necessarily, which is basically hiding in the soil from herbivores. We know there are differences in flowering time. Populations differ from one another, and there's variation within populations. So we're pretty confident that there'll be a huge amount of new both genetic and phenotypic variation. So I'll close with this slide, just to remind you that although this phase of it's really exciting and it's neat to go to Turkey and eat kebabs for 2 months and do the sequencing and understand the populations, but the real goal is to get this material into populations that can be phenotyped in an agronomic context. And to do that, we have to solve a problem that I told you earlier on, and that is the problem that the wild genotypes are very sensitive to climate. And they flower or not, depending upon the situation they find themselves in. And that's, the cultivated material is much less sensitive to environmental extremes. And we've identified 2 loci that control flowering time, probably a vernalization locus, and a second locus that controls a significant portion of the growth habit that differs between cultivated and wild. We'll construct a nested association mapping panel in which we normalize those 2 traits, phenology, flowering time, and growth habit to the extent that we can with those loci. And this will be our main experimental population. We'll select 20 individuals from the total wild material, because that's about all we think we can handle, using the environmental information and the genetic information, the genomic information to select those individuals. We'll cross them into 5 different elite genotypes that represent the major cultivation areas of chickpea around the world: Ethiopia, India, Turkey, Australia, and Canada. And we'll develop nested association mapping panels so we can more rationally try to assess phenotypes. And then we'll also develop a second type of population, and these are advanced back-cross integration lines, where the goal is to create, where these populations are basically 50% wild and 50% cultivated, except at the loci that we're fixing. These populations are essentially completely cultivated, except for a different segment of wild that's present in each of a different series of genotypes, and they're closer to being breeder-ready kind of material. So that gives you a sense of what we're up to. It'll take us a few years to develop the populations. We'll be able to start some functional analysis on the wild material itself before that, and we've started genomics. I should mention 3 people who are here. Varma Penmetsa from my laboratory at UC Davis is the project manager and lead scientist on both the NSF and USAID projects. Aznake Fikra, who is at EIAR in Ethiopia, and he's our project lead and a chickpea breeder in Ethiopia. The work that I described, but only briefly, on nitrogen fixation and, of course, sequencing the genome was conducted in collaboration with Rajiv Varshney. And then we have collaborators at each of these other universities in the United States, in Turkey, and in Australia. And I won't go into everybody's name, but those that are colored are the principal investigators, and the other individuals are people in their laboratories. So I'll stop there and answer any questions you might have.
Thank you very much. Are there any quick questions? Because we are running a little bit over time already.
Excellent work, Doc, and the collection will be really very useful to the community. Do you have any plan to sequence the existing reticulum and kinospermum collection along with your new collection?
Do we have any plans to sequence the genomes?
The old collections also.
Yeah, that's a really good question, Porin. So we have a flow cell, a couple of lanes of sequencing ongoing right now for RAD, because that's the first step, in which we've sampled the available wild material. It's not everything, but it's a representative fraction of the wild material, and it'll give us a sense of how that wild material maps onto what we have here, because it's important. And when we do the whole genome resequencing at greater depth, we'll include some of that material, because if we don't understand the relationship between what we've collected and what previously exists, then there's a big disconnect. So yeah, we will do that point.